Good morning, students. I'm Dr. Nadia. Today I'm going to discuss about the topic study of microbial structure. So in this microbial structure, we are going to talk about virus and its characteristics and its structure. So coming to the basic introduction of virus. Virus are tiny infectious agent that live and multiply only inside a living cells. It can also infect all types of life forms that can be animals or plants to microorganisms like bacteria and archaea. Generally, a complete virus particle with all its parts that can infect a host organism is called as a viron because without host it can't able to infect anyone. It can't reach to the particular organism. So this virus was first discovered in the year of 1898 by Dutch scientist Martinus Beijerinck. So how do this virus reproduce, or we can say as replicate? So you can see in this image. So generally virus, when it want to get to attach to body cell, the first step is attachment. Because without, so in our body cell there are many cells will be, many host cells will be there. Among that, which cell is very weak? So virus generally target particular weak host cell, because once it get the attachment, automatically it will get a easy entry gateway. So after first the main process is attachment. Then once it got attachment, it directly enter into the genetic material, and through the genetic material, the host cell cytoplasm, and then it will enter into the nucleus. So once it enter into the nucleus, what it will do, the substance of cell begin to strip off the virus outer coat of the protein. So once it's enter, this virus has a very and particular role, because whenever virus invaded to our cell, automatically it will start to rule the that particular cell to listen its own controlment. So that is the one of the main drawback about this virus. And here if you see, the nucleic acid in the center of the virus is released, then after then what will happen, it will get into the chemical manufacturing system. So after then the replication will be take place. The replication is a third step. So here what will happen, the viral genome is copied and its genes are expressed to make many viral proteins. Once the proteins are formed, then it start to assemble. So that is the fourth step. So new viruses are formed from the copies of genome and protein inside the host. Then finally, after assembling, it start to release the virus particle, and it will completely affect the other host cell. So coming to the characteristics of virus. So here general characteristics of virus are non-living nature, and it is a virus are considered non-living entities because they lack cellular structure and they cannot carry out metabolite process on their own. They only become active and capable of reproduction when inside a host cell. And coming to the genetic material, here virus contain genetic material either DNA or RNA, sometimes both are not. So genetic materials carries the instruction of virus to replicate and produce new virus particle, and protein coat. This is a capsid. It's because this capsid which protect that entire protein, that entire wall which surround the viral genetic material. And this capsid is mainly composed of protein subunit called capsule mesh, and at the same time envelope. So some viruses have an additional outer envelope composed of lipids which is derived from the host cell membrane. It acts as one kind of coat shield, as one coat shield. So through that the virus can be protect itself. So envelope viruses are often more susceptible to environmental factors. And host specificity. So viruses are highly specific to their host organism and may even target specific cell types within the host. This specificity is due to the interaction between the viral surface protein and host cell receptor. And finally, replication, because once it's invaded into the cell, it start to target other cell also to come under its control. So once the particle is released, so automatically the replication process will be takes place, then the entire cell will get hijacked. Then lack of cellular structure. Virus lack the cellular machinery found in living cells such as organelles and metabolic pathway. And insert outside the host. Viruses are generally inactive and inert outside the host cell. And they can survive in the environment but are not capable of metabolism or reproduction in this state. So coming to the structure of virus. So virus are tiny particles much smaller in size than bacteria. If you see there, they are wide variety of shapes like a popcorn, while other may have a complicated shapes of spider or even the Apollo lunar ladder. And if you see the diameter of virus is 20 to 300 nanometer and can be viewed only under a microscope. So in the absence of host cell, the virus cannot generally do any activities and it can't invade to the particular cell. And they cannot synthesize their protein due to the absence of ribosomes. So they cannot even store their own energy in the form of ATP. All viruses contain either DNA or RNA nucleic acid. Sometimes both nucleic acid also not be present, and it is also consists protein code which encases the nucleic acid called the capsid. And along with this, some viruses are even covered with a coat of fats and proteins, because which act as a shield for the virus. And whenever the virus infected, a particles called a viron is present outside the cell. In some virus, an envelope is made up of phospholipid layer and glycoprotein is present outside of the capsid. So you can see the structure in this slide. So how the protein is covered, you can see that lipid envelope and tegument and DNA, nucleocapsid envelope proteins, how it is tightly binded in that coat. So generally, here what is this part? If you see in the genomic or nucleic acid, this part contain DNA and RNA. So here the virus that contain the DNA protein is called DNA virus, and which contain RNA is called RNA virus. So as an example, Rio virus, when RNA is converted into reovirus, because whenever the RNA virus containing RNA genome, we used to call it as a reovirus. If DNA is converted into as a DNA virus, then it is called as papavirus. And capsid, which act as a protective protein coat, it comprises many capsomere which are arranged tightly together in the repeating patterns. And it is an impenetrable shell. Its main function to help introduce a viral genome into the host cell during the infection. The structure of capsid is what which gives symmetry to the virus. The structure can be, there are different types of structure like cubical, helical, complex or binal. So you can see in this image helical, polyhedral, spherical and complex. So for helical there is an example tobacco mosaic virus. For polyhedral adenovirus or spherical influenza virus. For complex bacteriophage. Let me see the difference of these types. First we will discuss about the cubical symmetry. So cubical symmetry is also called as isocadral symmetry. There are 12 vertices, 30 edges and 20 sides in this type. So each side is an equilateral triangle. So it is one of the most stable structure and found in human pathogen. For example, adenovirus, herp virus, tuberculosis virus. Another one is helical symmetry, also called as spiral symmetry. The capsomere nucleic acid are intertwined together to form a spiral tube. So here viruses are enveloped and all of them contain RNA proteins. So for this helical symmetry example is tobacco mosaic virus. And complex symmetry is nothing but they do not have any one particular type of virus structure. They will made up of two combinations of structure. Like for example, in the complex it's a nothing but a two structure which is involved like helical and cubical together present in one set of structure. So that is called as complex structure. For example, pox virus and binal symmetry. So binal symmetry, this is a type of complex symmetry where the virus structure have head and tails. So some of the most complicated structure of that or that of the bacteriophages that possess an isoscadral head as well as the helical tail. So those kind of structure we named it as T1, that is T2, T5, etc. Thank you.